What if some of the world's most advanced longevity solutions already exist in nature? What if animals that hibernate for months, survive oxygen deprivation, reverse organ damage, and resist fibrosis and neurodegeneration are already solving biological problems humans struggle to treat? And what if the future of longevity medicine depends not just on studying humans, but on decoding the biology of the most resilient creatures on Earth? Welcome back to Decoding Longevity. I'm your host, Dr. Raghav Sehgal, joined with my co-host, Dr. Macsue Jacques.
Hi, everyone. In our last episode, we explored cellular reprogramming and transcription factor therapeutics with Janine Sengstack. And today we're going to move into one of the most fascinating frontiers in longevity science, comparative biology and evolution-driven drug discovery. Our guest today is Dr. Ashley Zehnder, a veterinarian scientist, co-founder and CEO of Fauna Bio, a biotech company using AI, genomics, and evolutionary biology to discover new therapeutics inspired by animals that naturally resist aging-related diseases. Ashley, welcome to the podcast.
Yeah, I'm so excited to be on here and glad Janine introduced me to you guys. So it's great.
Well, Ashley, it is so exciting to have you, and honestly, so excited that Janine introduced you. But let's talk about sort of what you've been doing. In general, sort of most just drug discoveries sort of starts by studying humans or traditional model organisms, but Fauna Bio and the work that you've been doing at Fauna Bio is different in a way. You've been studying really cute hibernating squirrels and long-lived mammals,
Yep.
species with extreme resilience. What made you realize that evolution might be the best drug discovery engine?
Yeah, well, I come from a slightly unique background for a biotech CEO, and so do my two co-founders. I am a clinical veterinarian by training, and I did residency training in exotic animal medicine. So my patients were all birds, mammals, reptiles, with an amazing diversity of diseases and biology you can study in those species. And then have the opportunity to come down to Stanford and do a more cancer biology focused PhD. I was really interested in cancer mechanisms across species. And I was sort of struck by the lack of knowledge of these really highly trained, highly respected cancer researchers at Stanford to the breadth of diversity that we can see in these other species. And they were really only used to thinking about data from humans and then mice or rats that they sort of tried to give human diseases to. And so people in that context are really used to thinking about animals as a way to try to induce a disease that sort of looks like what happens in humans, without really understanding the fact that there are animals out there in nature who naturally can reverse diseases. And so we think at Fauna, we think about animal diseases in a very different way. We say, look, let's look out in nature where nature has solved these problems as a way to solve really challenging environmental evolutionary challenges that these animals face. We'll get more into hibernation data later, but let's learn directly from how nature, again, has design these solutions, and then let's link that directly to human diseases. So again, we kind of reverse the paradigm of how to think about animal models.
Yeah, I love that framing. I love that you don't actually have to induce or artificially create those diseases in the animals to then be able to study. You already, using what is out there. So I think it's fantastic. So it's very fascinating as well. So you said you talk about hibernation a little bit later, but let's talk about it. So hibernating mammals can endure months of metabolic suppression, muscle inactivity, low oxygen, and tissue stress, and then they emerge without having any fibrosis or any neurodegeneration or muscle wasting, which is not, a big problem when you stop moving. So how is that biologically possible?
Yeah, no, it's a great time to get into that biology. So hibernating mammals, and this is not just 13-line ground squirrels that we study and what our CSO studied for her PhD work, but actually a really broad swath of mammals that undergo this process of hibernation in periods of really resource scarcity. It's a way for the body to massively reduce metabolism and spare tissue from damage during this hibernation period. So everything as diverse as not only the rodents like the ground squirrels, but also lemurs that are primates, just like humans are, and Madagascar hibernate very very similarly to the ground squirrels, bats and bears. So there's a wide variety of mammals that can do this. What's interesting about the animals like the 13-line ground squirrel is that they'll take their body temperature down to almost freezing. They stay around 4 degrees Celsius. They're truly in a state of suspended animation. Their heart rate goes down to 5 beats per minute. Normally it's 250. Their oxygen level in their blood drops to about 10% of normal, about 90% reduction. Metabolism is down to 1 to 3% of normal. But that's actually not the most interesting thing. The most interesting thing is that they do that for about 2 weeks, but then they rapidly rewarm back up to normal body temperature, and that happens in only about an hour. So you imagine this really cold, hypoxic mammal getting a rush of warm oxygenated blood into most every organ of the body. It's basically what happens after a human heart attack or stroke. And obviously there are some differences, but there's a ischemic damage that happens in almost every organ in the body that then they have to repair that 25 times in a 6-month period. So evolutionarily, the animals that could do that have really hyper solved ways for that tissue to repair damage. And so there's ways to reduce reaction to oxidative species. There's ways to slow down inflammation, reduce fibrosis. There are ways to, at a whole organism scale, fundamentally alter metabolic rate. There are ways to recycle nutrients and build skeletal muscle, even though they aren't moving. So there are all these mechanisms that they've had to, because of their crazy physiology, derive solutions to over hundreds of millions of years that then now we can learn directly from for human diseases.
That's really exciting. And it oddly sounds familiar to like a natural rejuvenation biology that these organisms are practicing. And at Fauna Bio, what you're really doing is sort of taking all of that biology and sort of combining it with comparative genomics and sort of AI
Right.
and machine learning. So can you explain how are you really doing that? What is your platform like? How do you sort of go from sort of sequencing these resilient animals to identifying sort of therapeutics that sort of target humans?
Yeah. The biology itself is so cool, but if you don't translate it into something that can help, humans, then it kind of just sits in the science. So I'm glad you brought up regeneration as well, and I'll touch on that, and then I'll talk about the platform. So we see that oftentimes in many of the different tissues that we're studying, that these animals are able to sort of transiently reactivate this earlier, almost stem-like state. So in the heart, they're able to reactivate these stem-like factors in the heart cells, and then critically though turn them back off when they're not needed, because if you turn those factors on and kind of let them go unchecked, you can get tumors and other things. So it's this ability to, like I said, transiently turn on and off these kind of earlier stem-like factors that allow those tissues to regenerate, repair damage, and then sort of go back to normal. So there's all kinds of really interesting biology to study there. But aside from the really interesting biology, how we then find drugs. So our platform is called Convergence AI. It does what the name implies. It merges data from these naturally resistant species. We obviously work a lot with hibernating mammals, but we have worked with other species as well, like the spiny mouse, which is even more regenerative than the ground squirrel. And they're one of the few adult mammals that can fully regenerate tissues. So we take data from these emerging animal models where we have time points and tissues where we clearly see a disease reversal or resistant phenotype. So we'll take RNA sequencing, mostly single nucleus at this point, RNA sequencing from heart samples, from brain samples, from skeletal muscle at time points where we know that repair is happening. And then we merge that with human patient data. So for the heart example, we'll take data from the ground squirrel heart at these reparative time points. How does that compare with data from humans in heart failure? What's changing in the ground squirrel that's maybe going up in terms of gene regulation, maybe going down in humans with heart failure? Can we find these oppositely regulated networks? And then can we test the hubs of those networks and say, can we genetically perturb them, knock them down, or overexpress them in relevant human cell models is usually our first filter. So let's look at that in human cardiomyocytes. Can we protect them from damage? And then going into more traditional sort of animal models of induced cardiac disease. Oftentimes what we're also doing is looking at humans that may have natural variation in those genes that we're finding in the hibernation models. Is there a link to human cardiac disease? Is there a link to protective factors for heart disease for patients that naturally have mutated those same genes? We can also go directly from gene expression to compounds. And that's actually how we found our lead program, which I know we'll talk about as we go forward, is looking at mapping gene expression signatures to a data set of compounds that were used to treat human cells. This is a modified version of what's known as the L1000 data set from the NIH. And it allows us to make that leap from, like I said, gene expression data to compounds. And then we can take those compounds, test them in animal models and human cell models, and then modify those to be novel chemical matter to take forward for development.
That is very clever and very, very interesting. And I think going back a little bit, you're talking about translation, but I know that in Fauna Bio, you have over nearly 100 million years of evolution in data over there, right? From
Yep.
several different animals, from several different species.
Yep.
So that's an extraordinary amount of data in biology encoded in nature in itself. So I know that you touched base on some animals that you are working, but what were some of the most surprising examples that you have seen where those animals naturally resist diseases that are incredibly devastating in humans? Or, even how can you actually translate something that is happening from an animal to a disease in a human? Because I assume they're not exactly sometimes the same or clear. The mechanism behind might be, but
Mhm.
not the disease itself.
Yeah, no, exactly. I'm glad you asked the question. So I think what's been most surprising for us is how consistently we've been able to find these disease reversal networks in very different types of data sets and tissues. We started out really looking at the heart. It was a very well described that animals during hibernation are protected from heart damage compared to the time points in the summer. This is a model that's been studied in terms of the 13-line ground squirrel for decades in the hibernation community. So it's like if you're going to start somewhere, that's a low-hanging fruit in terms of extreme animal disease protection. But then going from there, we went into looking at the brain. And so looking at changes in neuronal metabolism, so parts of the brains that are able to stay functioning when everything else is shut down. They're sort of functionally brain dead when they're in hibernation. They have a completely flat EEG. The neurons in their brains retract. They're hyper-tau phosphorylated. But there are cells in the brain that are able to stay functioning, what's happening in those cells. And we found highly conserved genes that are responsible for fuel switching in the brain that translate very nicely to human cell models. And then going to the GI tract, a very different organ, very different structure, multi-layered, really hard to model in 2D. You need these 2 and 3 cell systems to really model it. Looking at things like immune cell migration and barrier function. So again, very different biological questions we're asking in the brain, very different biological questions we're asking in the heart. Also found pathways that then replicate in human cell models and with human genetics. And so I guess we sort of intuitively thought this was going to work. We've been sort of pleasantly surprised at how well in different organ systems we've been able to find druggable targets that meet our criteria across a very wide range of biology. And I think it comes down to asking the right questions of the right data and getting really clean data sets and comparing them in the right way. And that takes a lot of time. And I'm glad you brought up the complexity of the data because it really has been aided a lot in the advances in AI. And I'm not going to, beat that horse too much in this podcast, but we do need that because we have like one of the largest biomedical knowledge graphs put together with our animal data. It's we have a GNN now is trained on 1 billion model parameters, and it's just not something that a human brain can do. And then really being able to pull all of the different 8 applications that we use on the platform together into a concerted hypothesis around a target is also done using AI. And so the data sets have just gotten too large and complex where we've had to bring in these systems.
Yeah. And I think that's part of the challenge here that you guys have really solved, which is looking at this data in such a new and insightful way, which could not have been done in sort of some of the classical approaches that have been out there previously. I think I want to just kind of shift back the drive a little bit here to sort of the hibernating animals, which I find really cute. So hibernating animals, of course, they can repeatedly protect their brains, heart, and muscles through these cycles that you were talking about, which would be seriously damaging for humans as well, as you were talking about how in sort of these, if we sort of deprive ourselves of oxygen, which happens in cardiovascular disease, cardiovascular events or stroke, we of course have huge damages. How are we working towards sort of tackling this in other diseases like maybe Alzheimer's and so on and so forth? And I think I have a slightly futuristic question as well here. Do you think that could our understanding of what you're building right now lead to a future where Fauna Bio is translating something into cryopreservation?
Not that out there question. We get asked that question fairly commonly. I feel like some of the pathways that we're investigating certainly could be used to help tissues obviously tolerate longer, colder temperatures. We mostly work with mammal data at Fauna, and there's sort of a limit to what those mammalian temperatures naturally go down to. But I think there are some really interesting insights from species that are more like the turtles and deep hibernating frogs that their tissues do actually freeze during their hibernation time points. It's actually much more extreme. And so I do think there are solutions in nature to doing cryopreservation effectively. It's not something that we focused on a lot on, but absolutely, I do think there's lessons to be learned there. In terms of the brain and Alzheimer's disease, like I said, I was alluding to a little bit earlier, these hibernating mammals have some of the most dramatic examples of dendritic plasticity. So how the neurons change during the hibernation time course, being able to rebuild neurons and neuronal connections, which they lose every time they go through a hibernation cycle, which is crazy. They start to become tau phosphorylated, which is a buildup of a protein in the brain that is linked to Alzheimer's disease, and they clear it naturally, again, every 2 weeks. How do they do that? It's the same tau phosphorylation that we see in human disease, but they're clearing it rapidly, very frequently. So I think that there's a lot of links between metabolic disease, again, which we're working on with Lilly to find novel targets for obesity. There's a lot of links now between metabolic disease and, CNS diseases and neurodegeneration like Alzheimer's, and kind of this idea of type 3 diabetes, which is the effects of these metabolic diseases in the brain, right? So people like to try to break body systems up into distinct body systems, but really they all talk to each other, right? So I think there's a lot of links between metabolism, this dendritic plasticity I talked about, tau phosphorylation. There's a lot that we can dig into there, just on the CNS health side for sure.
Oh, that's incredible. And you, as you mentioned, you are using AI to find those drug targets. But are you actually developing, or are you using any of those drug targets that you might have found and translating into like therapeutics program? Have you actually started trials as well in Find Bio?
We're getting close. So it's actually a good time to be recording this podcast. We just finished a set of 60-day GLP tox studies on our lead program, which is a novel Fauna wholly owned program that we found with ground squirrel cardiac data. So Fawn 1083, we presented a poster at AHA last November, is a novel small molecule with a novel target for HFpEF and HFpEF with PH, which is particularly the HFpEF with PH. There are no approved therapies right now. It's a really difficult kind of heart failure to treat, but HFpEF more broadly affects about half of all heart failure patients around the world. So approximately 30 million people around the world have this kind of heart failure, which is much more prevalent in patients that have metabolic disease, so obese patients, and also more older patients, and also tend to be more female patients as well, and doesn't have a lot of great therapies. The long-term prognosis for these patients is quite poor. There's about a 5-year survival, 75% still alive, or less than 75% alive at 5 years. So there's a lot of need for novel treatments for this particular type of heart failure. And so we have completed a number of different animal efficacy studies and animal models of this type of heart failure. We have a lot of human genomics data suggesting that the target that we found with the ground squirrel data is important in patients. Like I said, and just completed the really critical safety studies to then be able to go to the clinic. So that'll be the next thing that we'll be focused on.
Super exciting. And so correct me if I'm wrong, this is a separate target from your antifibrotic candidate, which is the Fauna 1003, right?
Right. So actually, Fawn 003 was a tool compound that we found with the platform that is antifibrotic. Fawn 1083 is really the sort of next generation drug that's been optimized for cardiopulmonary disease. So we for that compound, see an improvement in mitochondrial function. We see an improvement in sort of the sympathetic nerve activity around the heart, so how hard the heart is working, and also see improvement in tissue fibrosis and tissue structures with animal models that we're treating with this drug. But yeah, it's kind of the 1003 2.0. Exactly. It's 1083. That's right. Yeah.
That's amazing.
This has been like really, really insightful. And I love how you're actually using those models. So I guess I have just a few more questions here. Instead of like asking on disease specifically and looking in the stopping decline, that's what is happening with aging virtually, right? Do you think we can answer such questions looking at those animal models that are actually, somehow living longer or even reversing some of those aspects of like decline? And do you think we can solve aging by looking at those animal models?
Yeah, I think there are a lot of great examples in nature, and where nature has multiple different times and multiple different ways derive solutions to the types of diseases and the types of tissue degradation that eventually do lead to aging, right? That what is aging except the sort of long breakdown of different body systems over time. So we can find ways to rejuvenate the stem cell niches in different body systems, the way that tissues can heal damage without inflammation or fibrosis, can improve the metabolic profile of cells. These are challenges that many different species have solved. I think what the bottleneck has been historically, and we're certainly not the first company to try to do this, but we're at a point in the science where the sequencing data is cheap enough to the point where we can start to get the same kind of robust computer interpretable data sets on a 13-line ground squirrel that we have been able to for a while from humans and, the 5 common model organisms. So the data sets are getting much more rich, much more complex, and much more able to be aligned with human data sets that people are typically using for drug discovery. So we're really kind of hitting a dawn of a new era with comparative biology. There are a number of other companies in the space, not just us. They're looking at these kind of natural solutions. And I think that there's this, I compare the comparative biology sort of field to where the longevity field was as a whole, maybe about 10 years ago, right? Like we're getting there, the data is getting better. There are companies that are translating the science, and there's becoming this momentum around why are we reinventing the wheel when if we just sort of poked our heads up and looked around, there may be these solutions that are out there waiting for us. But the data was just not really good enough to do robust drug discovery until about the time we started Fauna. We sort of came in right about the time that it was possible. And so we're sort of cracking that open. But like I said, there are more and more companies that are taking a look at the space really seriously and are intersecting quite naturally with the longevity field, because a lot of the diseases that these companies are tackling are related to DNA repair and metabolism and inflammation. And there's a natural overlap in those approaches.
Yeah. So, kind of a future-looking question here. What are you most excited about coming in the next 5 years? So there's of course a lot of AI-related changes that are happening in the space. There is sort of the alpha genomes and sort of those LLM models that are really exciting that are coming out, the foundational models. There's of course more data being generated in the context of comparative genomics. And there's of course other dimensionalities of epigenetic reprogramming and stuff that is also happening in parallel. What excites you the most, and what do you think is really going to be transformative in your opinion? And I think another question that I have for you before that is, who do you think should be who do you think should be the next person we should bring on this podcast to talk about longevity science?
Gotcha. Okay. What am I most excited about? I'm most excited about how technology is enabling the types of discovery that we and other companies in the space are doing, as I kind of alluded to before. And so I think there is going to be an explosion of new, rich biological data sets. I had to chuckle. I was at an AI conference, I want to say about a year ago, and they were talking about this sort of this data training wall. Like, we've trained on all the data that's available on the internet. I'm like, and I was just laughing because there was a biological panel there. I'm like, we're not even close to hitting a biological training wall. Like, we haven't generated the data enough to see that there is a wall, right? Like, there's just so much untapped biology in terms of asking the right questions. But I do think we're sort of slowly starting to get there. I think people are recognizing the value of complex, biologically faithful models really informs drug discovery in a much more useful way. And so I'm excited to see those data sets come out. I'm a little bit unclear the large foundation model for biology type approaches, like kind of a hammer looking for a nail, right? What are we going to use them for? And so I think for AI to be really useful, it has to be tuned on a specific biological question on the right biological data, right? So making sure the tools match the models. But I do think people are using AI in a much more refined way and recognizing the value of that complexity. And so I'm excited to just see people generating better, high quality data sets and then training the right AI models on those data sets. And I do think that field is maturing. So I'm excited to see where that goes. For us, I'm really excited to get our programs in the clinic. And I think when we see humans with heart failure improving in function and being able to exercise more for a drug that was discovered in the ground squirrels, like, amazing, right? Like, I think that will start to crack open the, like, oh my gosh, this actually works, even though we sort of known that now for a while. GLP-1 drugs didn't come from people, right? The original ones came from Gila monsters, right? There's a long history of finding really important innovations for human health and other species, and we tend to forget about that sometimes. So, but we have to keep reminding people. So I'm excited to see that. I'm excited to see more targets coming out of our obesity collaboration with Lilly. I think, we're looking at some really novel mechanisms that are not at all related to GLP-1 and how to modify human energy expenditure, how to spare skeletal muscle. And so I'm hoping we get some more of those into Lilly's pipeline, because those could get to patients with Lilly's firepower as soon as possible. So excited to see that also come out of the hibernation science. So I think it's a really exciting time to be in the field. There's a lot of energy, a lot of people working on different animal models, and a lot of tools available to help translate that data really quickly. Kind of along the same lines, to answer the second part of your question, if you haven't spoken to Dan Oliver and his team at Rejuvenate Bio, I think they're a really good one to talk to. Also in the aging space, also using animal models in a different way. I don't know if you're familiar with the company, but they test their therapeutics on natural models of disease. So they've worked a lot with canine cardiac disease, for example. They've really done a nice job of bridging the gap between animal health and human health. And they're also a really smart team to talk to. So I recommend them.
Yeah, Rejuvenate is doing some really amazing work for sure.
Yeah.
Just
Yeah.
thinking something. I think
Mhm.
as you get post phase 3 and you're deploying your first therapeutic in humans and you have your
Mhm.
advertisement, it'd be really nice
Mhm.
to have a squirrel being petted by an older patient saying, "That saved my life." uh,
They don't sit
li-
still unless they're hibernating. Maybe a
yeah,
hibernating squirrel. You can
maybe
pet the
hibernating.
hibernating ones. The,
Yeah,
the awake ones are like Tasmanian devils. Like you can't
yeah.
really hold them down. But maybe a
Lily
hibernate.
Couldn't have done that with the Killia monster for sure.
No, yeah. Those, yeah, the venomous lizards, not so
Yeah,
much. But
yeah,
the
yeah.
sleeping ground
You guys
squirrels,
can
we
do
can
it.
make that work.
You can make that
Exactly.
work.
That's right. Exactly.
Can always use the
Yep.
AI to pretend it's it's
It's
getting
true.
there.
It's true. Not as fun.
Yeah.
Oh, that's fun
Yeah,
for sure.
we did let Sanjay Gupta from CNN got to pet a sleeping ground squirrel. We
Oh,
had a
that's
CNN crew
amazing.
out at our team with our team at Oshkosh, and he was able to touch the sleeping ground squirrel. So
That's so cute.
That's cute.
yeah,
That's cute.
yeah,
Oh, thank you so much, Ashley. This conversation has really been amazing. I love how you're reframing the questions in a very different way, and so almost, shifting that paradigm that we have to always use the same models time and time again, and
yep.
sometimes not find the answers. So thank you so much for coming here and sharing a little bit of what Fauna Bio is doing on this field. And yeah, it's very exciting to meet you.
Yeah.
Yeah, nice to meet you guys as well.
Yeah. And to our listeners, we will link Ashley's work around Fauna Bio and several talks and interviews on comparative genomics. She gives amazing talks, by the way. I've heard her talk to many conferences. And in our show notes, if you enjoyed this episode, don't forget to follow, share, and leave us a review. Until next time, keep decoding longevity.